The eukaryotic cell cycle includes the mitosis phase, which is the process of cell division that consists of two processes, mitosis and cytokinesis. On the left-hand side, you can see photos of mitosis in cell from a fish embryo viewed by light microscope. The mitotic spindle, including asters, is visible in the metaphase and anaphase micrographs. At low magnification, you can see spindle fibers. Moving along to figure 19-1 above, in mitosis, the mitotic spindle segregates the duplicate condensed chromosomes into two daughter nuclei, and cytokinesis, the cytoplasm, divides to yield two genetically identical daughter cells. Now below you can see a figure. Between divisions, the cell is an interphase, which consists of S phase and two gap phases named G1 and G2. The cell continues to grow throughout interphase. In this slide, we will show the progression through the cell cycle shown by figure 19-32. The red bars indicate three important transition points in the eukaryotic cell cycle. At each of these points, conditions within the cell determine whether the cell will proceed to the next stage of the cycle or not. During late G1, the first control point occurs, suggesting that progression from G1 into S phase is a critical control point in the cycle. For instance, in yeast, the control part is called start, where they must have sufficient nutrients and must reach a certain size to pass through. Now, if we were talking about animal cells, the point is called restriction, which is influenced by growth factors, nutrients, cell size, DNA damage, and so forth. The dark green that you see on both sides of the figure and G1 and G2 indicate where the cell can exit from the cycle and enter a non-dividing state. Cells that have passed are committed to S phase, whereas cells that do not enter into GO and reside there for variable periods of time.
In this slide, two different cells from 2 points in the cell cycle will form together to form one cell with two different nuclei. So in part A, it shows how you have a cell from S phase and you have a cell from G1, and when these two are fused together, the S phase is activated. And in part B, it shows how you have a cell from M phase and then you have a cell from G1 phase, and when they're fused together, the M phase is activated. And the fusion of cells that can be brought upon by like viruses or polyethylene glycol, and which is like by a brief electrical pulse, which causes plasma membranes to destabilize, and how the cells fuse together. In this slide, cytoplasm was extracted from a mature frog egg cell and injected into an oocyte, and the oocyte was still able to go through meiosis and become a mature egg cell. This procedure could also be used to detect or isolate MPF, because not only does MPF induce meiosis, but it also triggers mitosis when it is injected into the fertilized frog eggs. In figure 1935, you see how the MPF activity, it stays constant throughout, a constant up and down motion, but the mitotic cycling, it constantly rises from G1 to S to G2, and then at the end of G2, it triggers the onset of mitosis, and when it gets to mitosis, it just drops down very fast, and that's the decline of the mitotic CDK, and it prevents another mitosis from occurring.
This slide shows the early stages of mitosis and the mitotic process of the phosphorylation and dephosphorylation of CDK cycling, as well as the full mitotic CDK cycle. First figure on the slide shows the mitotic CDK and the mitotic cycling binding together, initially forming the inhibiting kinase, which includes 2 ATPs and 2 ADPs. The next stage would be the active kinase, which would be 3 phosphates, 2 inactive, 1 active inside the mitotic CDK. The mitotic CDK cycling is not active until the 2 white phosphates on the figure are removed and left with the one yellow active phosphate, and then you have an active mitotic CDK cycling. The second figure shows the full cell cycle and where the mitotic CDK cycling is located and where it takes place at in the cell cycle, and it takes place at the end of G2 where it transitions to M, the G2M transition as the figure labels it. Along with that, the mitotic CDK cycling, you have the activation of, you have the nuclear envelope, the breaking down, the chromosomes condensation, and the third part is the mitotic spindle forming and the target proteins degrading. This next slide shows the anaphase promoting complex and the mitotic spindle checkpoint. In the top half of the figure, it shows the anaphase promoting complex targeting the saccharin and the mitotic cycling for degradation. The destruction of the saccharin allows the separase to cleave to the cohesins that hold the sister chromatids together, thereby initiating the anaphase. The second step of the top half shows the degradation of the mitotic cycling. It depresses the mitotic CDK activity, leading to cytokinesis, chromosomes decondensation, and the nuclear envelope reassembling. In the bottom half of the figure, it shows the mitotic spindle checkpoint. In the mitotic spindle checkpoint, it prevents anaphase from starting until all chromosomes are attached to the spindle. As you see on one half, there is no spindle, but when there is no spindle, the MAD and BUD proteins are attached to an anaphase promoting complex. The checkpoint is left on by these MAD and BUD proteins attaching to the anaphase promoting complex, or the red area of the anaphase promoting complex, which is called CDC20. This prevents CDC20 from activating the anaphase promoting complex. After all those sister chromatids have spindles, then the MAD and BUD proteins are not formed, the checkpoint is turned off, and the anaphase promoting complex is free to initiate anaphase.
Okay, so now we're going to talk about how G1 CDK cycling regulate progression through restriction points. So restriction points are like checkpoints in the cell. So if things aren't going right, then they won't pass the restriction point and go on to the next phase. In this case, we're talking about the restriction point located in the late G1 phase. And like I said, it's going to determine whether the cell's able to move on to a different phase. So in this case, the S phase, and continue with the cell synthesis, cell division, and going on with the rest of the cell cycle. And in this case, we're going to talk about the RB protein. The RB protein controls expressions of genes whose products are needed to go through restriction point into the S phase. So yeah. Okay, so the G1 CDK cycling regulates progression through the restriction point by phosphorylating the RB protein. So prior to being phosphorylated by the G1 CDK cycling, the RB that's shown in purple is going to bind to and inhibit the E2F transcription factor that's shown in the green on your figure. The E2F would normally activate transcription needed for the S phase, but as long as it's binded to the RB protein, that's not going to happen. It's just going to remain inactive. So when the cells are stimulated by growth factors, a signaling pathway is triggered. This pathway is called the RAS pathway. Once this pathway is activated, as shown in the middle part of the figure, the production and activation of a G1 CDK cycling complex occurs. That's the pink and peaches figure in the middle of the screen. This is going to use up ATP and convert it to ADP. This complex catalyzes the RB phosphorylation. Once RB is phosphorylated, it no longer is able to bind to the E2F, which is shown at the end of the screen. The RB has the two phosphates connected to it, and then the E2F is no longer binded to it. So the transcription factor is now able to transcribe these genes that they weren't able to when it was binding to the RB. So it'll go through gene transcription, you can have mRNA translation, and then move on to the S phase. And then mitosis, well, the M phase, the RB protein is going to be dephosphorylated. So now the E2F can be inhibited once again. Okay, checkpoint pathways are also used to monitor chromosome spindle attachments, completion, DNA replication, and DNA damage. So a lot of things can go wrong with this cell. With the cell cycle. For example, an aneuploidy, literally meaning not a good ploidy. Chromosomes haven't been properly attached to the spindles yet, and they start moving towards the spindle poles. And of course that can happen because then you have too many copies of one type of chromosome, or sometimes a chromosome might not even copy at all. So to prevent things like this from happening, checkpoint mechanisms are used. Now these are similar to restriction points in the way that they monitor what's going on in the cell and determine if they can move on to the next phase or not. But checkpoints have they have the opportunity to actually completely stop the cell cycle from going on. There are different types of checkpoints. The checkpoint known to prevent aneuploidy is known as the mitotic spindle checkpoint. The way this works is basically the unattached kinetic cord sends out the signal that says, wait, like I haven't attached yet. So it's going to inhibit the anaphase promoting complex. Another type of checkpoint is the DNA replication checkpoint. This monitors DNA replication in the cell. So it's going to prevent the cell cycle from moving on to the G2 phase if DNA replication hasn't been completed. The third type of checkpoint mechanism is the DNA damage checkpoint, which is going to stop damaged cells from going through with the cell cycle unless the damage has been repaired. They do this through CDK cycling complexes. Late G1, S, and late G2 phases all have DNA damage checkpoints. So the figure on the screen shows what can happen with the damaged DNA at the DNA damage checkpoint. The damaged DNA activates the ataxia telangiectasia mutated protein kinase, also known as ATM, or the ATM-related ATR protein kinase. This activates the checkpoint kinases that phosphorylate the P53, which plays a very important role in the checkpoint mechanism. Now, normally P53 would interact with MDM2, shown in the red, and P53 would be marked for degradation, but since this interaction was blocked by the phosphorylation of P53, this does not happen, and one of two things occur. So marked by number 1 on the left hand side of the figure, phosphorylated P53 binds to the DNA and activates transcription for P21 protein. P21 stops cell cycle progression by inhibiting CDK cycling activity. If this happens, the RB protein cannot be phosphorylized and the cell will enter cell cycle arrest at the restriction point. Now on the right side of the figure marked by number 2, this shows what happens when the gene cannot be repaired. P53 will be inactive. P53 will activate genes coding proteins that trigger apoptosis. Now PUMA, which stands for P53 upregulated modulator of apoptosis binds to and inactivates BCL2, which is an inhibitor of apoptosis. Now, when cells like this are exposed to carcinogenic agents that cause DNA damage and the p53 is stimulated to go on and trigger cell cycle arrest or apoptosis, damaged cells are prevented from proliferating. Sometimes this whole process is missing and the pathway can lead to failure of apoptosis, which can then contribute to cancer development.
Figure 1941 is basically a figure of quote-unquote cell cycle regulation machine, and it talks about the cyclins that bind to CDKs to create a CDK cyclin complex that's going to trigger the cell cycle transition points that allow like the G1 phase to go into the S phase, and then S phase to go into G2, then G2 to go into M phase. So the influence that it has is basically to continue the cell cycle to progress. So it is broken down into the first part where the G1 is, and then it goes on to S, then G2, then the top part is where the actual, mitosis is actually occurring. So starting on the right side of the figure, we see that this is where G1 is happening. So G1 CDK, G1 cyclin is going to bind with each other by kinases that are going to add phosphates to them, which are going to activate them. So when they go to that first restriction point, that's going to allow them to continue on to the S phase. And at this point, G1 CDK is going to go all the way around, to the back to where it started, so that it can continue to bind to other G1 cyclins that are going to be added, because as this process continues, G1 cyclin is degraded. So then the mitotic CDK that's to the upright of the figure is going to move past all of this, and it's going to be activated when if you look to the up left of the figure, you see that it gets activated when it is binded to the mitotic cyclin. So the mitotic CDK and the mitotic cyclin are binded together essentially by two phosphates that are inactive, but it only gets activated when the active phosphate gets added to it. So two inactive and then one active, and then the phosphorylated complex is inactive, and it remains that way until the two inactive phosphates are removed. So then when it gets to the G2-M transition point, that's, as before, is going to allow them to go into the metaphase anaphase transition. So then the anaphase promoting complex is going, well, in this figure they are inactive, but when they are active, then they're going to allow them to go through, and then this process is just going to be able to continue to go on. And these checkpoint pathways, they monitor the cell for DNA damage, DNA replication, and chromosome attachment to the spindle. And as the degraded mitotic cyclins are taken away and the degraded G1 cyclins are taken away, it allows the process to just continue. This next figure talks about the stimulatory growth factors that activate the ROS pathway. So this basically just talks about the activation of the ROS protein by binding to the growth factor. Like the growth factor binds to the receptor, and then the receptor activates cytoplasmic protein kinases, and then it goes on to activating transcription factors, which eventually end up synthesizing E2FK, cyclin, and CDK. So here at the top, we see the growth factor binds to the receptor that's in the plasma membrane, and this is going to lead to the activation of the ROS. You see to the right of the figure, the ROS is getting activated by the GDP. It's going to attach to it to make the ROS active. So after that happens, this is going to phosphorylate the activation of the RAF, and RAF is a protein kinase. So after all of this happens, this in the second part of the figure, there's like the top part and then there's the bottom part. On the bottom part, when all of this activation has already happened, you see where it says step 2. That's where the protein kinase, RAF, is going to go and phosphorylate the serine and 309 or threonine in the MEK protein kinase. So that in turn is going to phosphorylate the threonine and the tyrosine in MAPK. MAPK stands for the mitogen activated protein kinases. So the RAF goes to phosphorylate MEK. MEK, after it gets those two phosphates that you see there in yellow, it's going to phosphorylate MAPK, and you see it has its two phosphates attached to it. And then when you see part 3 is where it can actually go into the nuclear membrane. Inside the nuclear membrane, that's where you see there's 4 different transcription factors. There's there's 2. I'm sorry, ETS and JUN. So those get phosphorylated, and then they activate transcription of the early genes, which are those 3: the JUN, FOS, and the MYC. That's going to activate the transcription of delayed genes. So you see at the end of this whole process, at step 4, there's 3 main things that we have to focus on. There's the E2F transcription factor that is going to control the entry of the S phase. And then there's also, like we talked before in the previous figure, the cyclin and CDK. Those bind together to make a complex that's going to phosphorylate the RB protein, which is also going to trigger the passage from G1 into the S phase. Here in figure 19-43, we see the stimulatory growth factors that can activate the PI3K ATK pathway. So a little side note is that the activation of the ROS pathway that we just talked about can activate this pathway at the same time. So the ATK suppressors can lead to apoptosis, which is when the quote-unquote bad protein gets phosphorylated and activated, and that's you see to the left of the figure. So the left of the figure shows basically what happens when ATK gets phosphorylated and then it goes to the bad protein. And then on the right side of the figure, you see what happens after ATK gets phosphorylated, and then it goes to RHEB, which then goes to activate TOR. And then TOR is going to promote cell survival and proliferation. So the first, outcome of this pathway is to activate and phosphorylate ATK. So you see at the middle, after the growth factor has been attached to the PI3 kinase, activates it, adding two yellow phosphates as you see there, and it can either go left, that leads to apoptosis, or it can either go right, that leads to cell growth. So if it goes left, then that protein, that bad protein, ATP is going to give it a phosphate. ATP is going to turn into ADP. Phosphate is activated and it inhibits the cycle, killing the cell. Right side, RHEB gets activated, TOR gets activated. Cell growth is, I don't want to say continued, but like cell growth is allowed and it continues, and cell survival and proliferation is promoted.
The following figures are about apoptosis and the growth and death of cells. The cell illustration shows chromosomes condensing and the cytoplasm shrinking. In the long run, the nucleus and the cytoplasm become fractured, ultimately blending numerous bubbles. Lastly, the fragments of dead cells are then ingested by the phagocytic cells. When the epithelial cells come into contact with another epithelial cell, it forms flat sheets. The flat sheets are held together through tight junctions and gap junctions. To ensure apoptosis, the cells exert a kinship with one another and weld together. And in slide or figure D, this basically is a single dead cell with many apoptotic bodies, as shown. Apoptosis is triggered by death signals / withdrawals of survival factors. The ligand binds to the death receptor. This process clusters the initial procapsase. Then the initiator procapsase becomes activated. Once the initiator becomes activated, the executioner procapsase initiates apoptosis. The survival factor is no longer present, so death promoters accumulate by counterbalancing the BCL2 on the mitochondria, acting as an inhibitor. The result is the release of cytochrome C. When cytochrome C and the adapter form a complex, the end result activates the initiator procapsase, which is procapsase 9. The activated initiator procapsase activates the apoptosome, which then activates the executioner capsase. The DNA end result is damaged, which leads to the production of p53 protein. In the following figure, it compares normal cells to tumor cells. Normal cells rise towards the outer surface, changing shape and losing capacity to divide, while the tumor cells rise towards the outer surface but still maintain their ability to divide. This is all shown in the lighter area. Normal cells maintain balance in the basal layer when a new cell is formed and the old cell dies, meaning that the lighter layer is being formed and the darker layer is losing the capacity to divide. In tumor cells, they have no balance to maintain the growth of the cells, which leads to the increasing of dividing cells.
All right, so this figure is showing two experiments being compared, as well as the importance of angiogenesis, which is something that causes cancer cells to grow.
Angiogenesis is the growth of blood vessels and balances the regulation that tumors need. In order for angiogenesis to proceed, it has to overcome the inhibitors. The inhibitors are angiostatin, endostatin, and thermospondin. The photo on the left is illustrating the cancer cell on a thyroid gland where the blood supply is very low. So as a result, it's not going to grow too much
All right, so this figure is showing how cancer begins, and it takes several steps before cancer becomes cancer. So the first step is that a DNA mutation must happen, and that's generally going to happen during the initiation stage. So after that stage, the next thing would be for that initiated cell to get turned on, and that will allow the mutated cell to have a longer lifespan, which is known as the proliferation stage. So by now we're at the promotion stage, and the last stage for the cancer cell is that the tumor is called tumor progression, and it occurs when mutations and changes in gene expression produce a variety of cells that show better growth rate or other strong properties, which allow certain cells to grow longer than the other cells, and ultimately those cells will become the dominant cell within that tumor. So in a nutshell, the way that this process goes, the first step is the cell is mutated. After the cell is mutated, the mutated cell has the advantage. So it's going to go ahead and outlive the other cells. From there, the mutations are going to produce a variety of cells that are more fit than the other cells. After that, those fit cells will become dominant cells. And once those dominant cells are there, they'll reproduce and progressively change over time. And that's how cancer cells begin.
Proto-oncogenes are converted into oncogenes by several different mechanisms. Proto-oncogenes arise by mutations from normal cellular genes. Oncogenes is a gene that can trigger the development of cancer. There are different mechanisms to convert proto-oncogenes into oncogenes. One is point mutation. The second is gene amplification. The third is chromosomal translocation. The fourth is local DNA rearrangements. And the fifth is insertional mutagenesis. On the top left-hand side, the figure is described as point mutations, which is A. It creates an oncogene coding for an abnormal protein that codes for a single amino acid from the normal protein produced by an oncogene by a single nucleotide substitution. B is gene amplification. It is the excess production of a normal protein because there are multiple gene copies being made. Chromosomal translocation is when chromosomes segments are moved from one chromosome to another. Local DNA arrangements causes abnormal proteins when insertions, inversions, deletions, and transpositions occur. Insertional mutagenesis occurs when inverted viral DNA is integrated into the host chromosome and causes the proto-oncogene to make too much protein. The bottom left picture is described as chromosome 8 contains normal MYC genes that frequently exchange with a segment of chromosome 14. The reciprocal translocation places the normal MYC gene adjacent to an active region of chromosome 14. This region contains genes used for coding antibodies. When the MYC gene is close to an active antibody, activation occurs leading to the overproduction of MYC protein. Oncogenes code for components of growth signaling pathways. Oncogenes have been grouped into 6 categories. One is growth factors. This is the cell can stimulate its own proliferation and receptors. Oncogenes may code for mutant receptors that permanently activates tyrosine kinase activity. Plasma membrane, GTP binding proteins. This is RAS, the most common type of genetic abnormality. Non-receptor protein kinase. is a protein that is not an intrinsic part of the cell's surface receptor. Transcription factors. This is non-receptor protein kinase, trigger changes altering gene expression. Cell cycle and apoptosis regulators. It accumulates proliferating cells, inhibiting apoptosis. The top left figure is described as A normal receptor activates by binding the growth factors to the receptor. This causes tyrosine kinase to be activated, causing each receptor to autophosphorylate. B is the mutant receptors have permanently active tyrosine kinase. The growth factor is missing, which makes this receptor constantly active. And C is illustrating amplified oncogenes produced endless normal receptors. This leads to excessive receptor activity. The bottom left figure is explaining, it has a chart that explains the different oncogene types and their proteins. Tumor suppressor genes are genes whose loss or inactivation can lead to cancer. Normal cells and cancer cells are fused artificially. Nuclei that contain chromosomes from both cells are formed during cell division. During cell division, some chromosomes are lost. Cancer cells demonstrates uncontrolled growth. P53 tumor suppressor gene is most frequently mutated gene in human cancers. The top left-hand corner, the figure there illustrates normal cells fuse with cancer cells. The hybrid cells do not intermediately form tumor cells. After an extended period of time, the hybrid cells convert to uncontrolled cells. Tumor-forming behavior of the original cancer cells. This is due to the loss of chromosomes that contain tumor suppressor genes. This gives the new uncontrolled cancer cell a new phenotype and new capabilities. For the figure below that, is illustrated as when a parent passes on the defective RB gene, the mutation will be in every body cell. If a good RB gene mutates in one cell, the RB gene becomes defective, and mutations become copied into other cells. But in families that do not carry the genetic makeup, the children are born with two good copies of RB gene. The cells divide, but it is very unlikely, that both copies of RB gene undergo mutation in the same cell. For the right-hand figure, it is explained as HPV has E6 and E7 proteins. The E6 triggers ubiquitin to attach to P53, which is the tumor suppressor gene. This contributes to the destruction of P53. After E7 binds to RB, which doesn't allow cells to stop at the restriction point, the E6 production happens.
The development of cancer happens when an accumulation of mutations that involve oncogenes and tumor suppressor genes occur. The most common mutations involve proto-oncogenes that turn into oncogenes in addition to tumor suppressor inactivation. In other words, tumor suppressor genes allow uninhibited cell growth, creating oncogenes. Genetic instability means mutations. This causes defects in DNA and defects in chromosome sorting.
When defects in chromosome sorting, with every successful mutation, abnormal cell behavior can be associated to that. In figure 24-21, this illustrates an organized step-by-step development of colon cancer. In step 1, you can see that normal cells have an APC mutation, which begins in early benign tumor. In step 2, the early benign tumor undergoes a KRAS mutation and then is progressed to an intermediate benign tumor. And then in step 3, the intermediate benign tumor undergoes an SM84 mutation and is progressed to a late benign tumor. In step 4, the late benign tumor undergoes a P53 mutation and is progressed to localized cancer. In step 5, localized cancer can undergo other mutations and epigenetic changes and allow the cancer to invade or metastasize. Carcinogenesis is a series of events that takes normal cells and it converts it into cancerous cells. There are 4 main types of causes of these cancers. Like I said, the 4 main causes of cancer is radiation, chemicals, infectious agents, and heredity. These trigger mutations in the DNA that create oncogenes and inactivates tumor suppressor genes. These changes create genetic instability, and when genetic instability is accrued and epigenetic changes occur, they develop into hallmarks of cancer. Hallmarks of cancer are self-sufficiency in growth signals, which is continuous stimulation for the cell to grow. The second one is unsensitivity to anti-growth signals. These anti-growth signals, they avoid each other. The third one is evasion of apoptosis, and this disrupts the P53 protein and its ability to trigger apoptosis. Number 4 is limitless replicative potential. And the 5th one, sustained angiogenesis, which is creating its own blood supply for the tumor growth. And then the last one is tissue invasion and metastasis. In terms of treatment, screening, and diagnosis, there are a few differences between benign tumors and cancerous tumors that are picked up by just some general characteristics. In table 24.3, you can see that there are some basic generalizations of these disease symptoms. In the benign tumor, you have the nuclear size is small, their ratio is low, the nuclear shape is regular, the mitotic index is low, tissue organization is normal. They are well differentiated and well defined. So you can see on the bottom of the picture of the benign tumor that it's very circular, it's very defined, it has a definitive shape. It's smaller in size. And then in the malignant tumor, you can see that it is a little bit larger than the benign tumor. It has a high ratio of nuclear to cytoplasmic volume. It's pleomorphic. It doesn't have a defined regular shape. Its mitotic index is high. Its tissue organization is none. It doesn't have an organization at all. It's disorganized. It's differentiated. It's not differentiated at all. It's very poorly differentiated and very poorly defined. So you can see the picture on the malignant tumor side. It's kind of crazy in shape. It doesn't have, an exact shape like I said earlier, it is pleomorphic. So those are the main differences in a generalization, when you're looking at it, when a doctor's looking at it. To get a real definitive diagnosis, it does require a biopsy. Sometimes it's, removed surgically by a small tissue sample. A lot of times they'll send it off to the lab for further, recognition to make sure to see exactly what it is. And that's it.